Hello everyone and welcome back to Microbiology. Today we'll be discussing Chapter 9, Microbial Growth. Previously we discussed the function of the glycocalyx, the sticky layer on the exterior of the cell wall. When well organized and tightly bound to the cell wall, it is referred to as a capsule. However, when loosely bound, this layer is endearingly referred to as a slime layer. Can you recall a major structure that the slime layer can lead to? Hopefully some of you thought of a biofilm. Here we can see the slime layer as a loosely attached glycocalyx. This allows the slime layer, the cells in a slime layer to attach to each other, attach to surfaces, form biofilms, and also increases their virulence by protecting them from removal or digestion or attack by our immune systems. Here is an image of the steps required for biofilm formation. Biofilm formation begins with the attachment of planktonic cells. Planktonic comes from the Greek word planktos, meaning drifter or wanderer. The first step is a reversible attachment of these planktonic cells, which takes just a matter of seconds. The second step is when the first colonizers become irreversibly attached, which can take seconds up to minutes. Eventually, naturally, this will lead to growth and cell division, which can take hours or days. And then in step 4, we see the production of exopolysaccharides, otherwise known as EPS, and formation of water channels. And again, this can happen in a matter of hours or days. And then step 5 is the attachment of secondary colonizers and dispersion of microbes to new sites, which can take days or months. Let's review step 4, the production of exopolysaccharides, which are sugar polymers that exist outside of the cells, and the formation of water channels. So who can guess what the purpose of water channels or channels in general would be on a biofilm? So if you can imagine a mass of cells starting to accumulate here, it becomes hard to transfer nutrients inside to the cells in the middle here or get rid of waste products. So as the structure grows, microenvironments begin to form, such as an anaerobic environment, so those are environments that are very low in oxygen, and then a buildup of waste products from one organism can eventually become a resource for another. So these channels and pockets serve for a method to provide the circulation of such chemicals and nutrients. And so the question can be asked, how and when do cells know when to stop development or to properly respond to biotic changes to their environment? Microbes do so through the use of something called quorum sensing, which is the regulation of cellular activities based on cell density, or the number of cells per a given volume or area. And this enables the detection of cells and critical thresholds of populations, or a quorum. The way it works is cells produce what are called autoinducers, which are small and diffusible molecules that are detected by cells and initiate a cascade of genes that affect cellular activity useful to a population or cell at that given density. So cells produce these small molecules, and as the culture becomes more and more dense, these molecules then initiate certain genes to be expressed, which give certain functions to the cells that are useful in those specific conditions. Those specific conditions meaning a very high cell density. So when the microbes have essentially grown quite a bit. So that brings us to the definition of what is microbial growth, which has a very specific definition. It is the proliferation of microbes through cellular division. So specifically, this is the process of growing larger in number, not larger in size. So more members of a population, not just bigger members. Unlike with humans, you might think of human growth as you growing up from a baby to an adult. Microbial growth is specifically about population growth. So let's take a look at what replication looks like for a bacterial cell. So bacterial cells replicate through binary fission, which is the splitting in two, essentially. So here we see this bacterium. It used to be one, and then it's split into two different ones. So you need to have replication of different components of the cell, like DNA replication. Then you have a cell elongation, so this coccus becomes more ovoid in shape. From then on, the cell is triggered into start making two different cells, and you get this formation of a division septum. So this right here is the inward building of the cell membrane and cell wall. And eventually, when this process is complete, you can have cell separation into two different cells. The process of creating new cells is called cytokinesis. So this is the actual cytoplasmic. Remember, the cytoplasm is the solution inside of the cells. So cytoplasmic separation would be separated by this membrane here that's in cell wall that's created, bringing about the division of the two daughter cells. This involves a whole complex of different types of proteins, but probably the most important here is the FITZ protein, which is very important in creating what is known as the Z ring on the inner membrane. So the Z ring is essentially a ring of proteins that are assimilating the cell wall. It's anchored by these FITZ binding proteins, and it establishes a protein complex, and which is called, cleverly, the divisome. And the divisome then activates the elongation of the peptidoglycan layer and cell membranes, leading to cytokinesis. You don't have to memorize this, but if it helps you remember FITZ, FITZ means filamenting temperature sensitive mutant Z. And this is important also for an identification, because you must be aware of the septum, as you will see its presence in some cells that might appear to be bacilli but are actually dividing cocci. There's another method for replication called budding, which is another asexual, a meaning without, so asexual reproduction, which is exhibited by yeast cells and some bacteria, in which reproductive division yields a parental cell and a smaller daughter cell. So here we can see the parental cell right here, and then this cell is budding off from the parental cell, and this creates it's not binary fission. It doesn't create two equal cells entirely. It creates a larger adult mother cell or parental cell and a much smaller daughter cell. And here is an electron microscope of that image as well. And right here, if you notice these circles, these are what are known as budding scars. So these are where the process of budding has occurred once before. And in general, you will note that there's an asymmetric dividing of these cells. You don't have a typical septisome that forms between them. Okay, so for growth, there are several requirements. Some of these are abiotic in nature, and some of them are biotic. We have to worry about things like temperature, the pH of the solution or medium they're growing on, the osmotic pressure, the oxygen levels, and the nutrients. So, with all of these parameters, you have specific components to their requirements. So, for every species of microbe, it has a specific temperature range. So that's the range in which the microbe can grow in. So we can see on this image here, it might be around maybe 2 degrees Celsius or so, or maybe a little bit higher, all the way up to, I don't know, it looks to be maybe 46 degrees Celsius before you have a specific growth rate of 0. A specific growth rate of 0 is no growth at all. Usually most microbes have a optimal growth of around 30 degrees Celsius, and that's because the conditions here on Earth, we have hot and cold, but 30 is kind of a medium between all of them. And a lot of microbes are actually associated with animals which have an elevated body temperature. So the other thing you're going to note on this graph, besides just a range, is that there is an optimal temperature. So that's when you have the highest growth rates. So for this organism, it appears to be about 40 degrees Celsius. So for this particular breed of E. coli. Although it looks like a lot of them are actually behind, so maybe this curve isn't a perfect fit for this growth curve. So it looks like maybe the best growth for some of these samples taken is actually a little bit under 40 degrees Celsius. And this is a strain of E. coli. Let's take a look at some other examples. So with E. coli, it says the minimum growth is about 4 degrees Celsius. The optimal growth typically for E. coli is 37 degrees Celsius. And then typically for E. coli, the maximum growth is 45 degrees Celsius. And this can vary a little bit with different strains of E. coli. So just like you have different strains of fruit trees or animals that that are grown for animal husbandry and plants and things like that, you can have different strains of bacteria as well. The other thing you'll notice is that this chart, this graph, once you reach the optimum, it tends to drop off precipitously. And the reason for that with temperature is once you get so hot, as you get warmer, things in the cell tend to be more and more active. But at certain points, it's just too hot that it leaves it just cell death and things just kind of fall off a cliff. So you can see this is an increased level of the ability for it to be enzymatically active. So things happen a lot quicker in the cell, and so you get quicker cellular division because you get the faster production of energy, the faster bioaccumulation of certain nutrients and things like that. Once you pass that certain temperature, you start denaturing proteins, structures start breaking down in the cell, and so it just crashes. So you kind of have like a slow increase, and then it reaches the optimum, and then it falls precipitously like that. So all microbes can be categorized based upon their ranges that they like to live in and their optimums. And scientists can group these into 4 general categories. So we have the psychrophiles, the mesophiles, the thermophiles, and the hyperthermophiles. And you won't necessarily need to know the very precise value, but if I give you a temperature, you should be able to tell me if that is an example of where you'd find those microbes growing. You should be able to tell me if it's a psychrophile, a mesophile, a thermophile, or a hyperthermophile. So let's begin with mesophiles. Most of these bacteria we deal with are mesophiles. So mesophiles have an optimum of around a little bit more than 30 to a little bit over 40. Mesophiles are the type of organisms that inhabit and are part of the microbiome. You can think of E. coli, for example, is right around 37 degrees Celsius. So it's going to lie in the mesophile range. And I'm not going to give you a question that's right on this interface here and really try and test your ability to tease out, 21 degrees Celsius, is that a psychrophile or mesophile? But if I give you a good representation somewhere under the curve, you should be able to tell me what it lies in. So 37 degrees Celsius is the temperature of the human body. And room temperature is somewhere around 22 to 25 degrees Celsius. So like I said, most of the organisms we study are mesophiles, and so that falls, within the room temperature to elevated body temperature for human beings. The one below that are called psychrophiles, which means cold loving, and they are types of organisms that might live in very cold environments or grow at the bottom of the ocean. And these can actually grow in sub-0 degrees Celsius temperatures, so 0 is when water freezes in Celsius, all the way up to a little bit under room temperature, 20 degrees Celsius. So we often refrigerate our food in order to reduce the growth of mesophiles. But we're not going to really fully stop some things like psychrophiles. We might slow them down a little bit in food refrigeration, but we're not going to fully prevent them. So next on the list are thermophiles. So thermophiles inhabit pretty warm temperatures. Thermophiles can grow, above the temperatures really seen in the human body, all the way up to about 80 degrees Celsius. So you can find these around areas like hot springs. And then next you have hyperthermophiles. So hyperthermophiles can again inhabit hot springs, and they're just a lot closer to incredibly hot temperatures. In fact, they can grow under temperatures that at 1 atmosphere the water would actually boil away. So they can grow in areas where they experience high temperatures and pressures, and can actually grow in temperatures above 100 degrees Celsius, which is boiling point at sea level. Now, next on the list is pH. First, let's talk a bit about what the pH scale is and what pH is. The simplest way to define pH is a unit of measurement of the concentration of protons or hydrogen cations. Here, protons that are positively charged in an aqueous solution. So protons are just hydrogens without any electrons, and that's why they're H+. And these are very important for chemical reactions in biology, because protons are very common with aqueous solutions or water solutions, as they make up 2 out of every 3 atoms in water, H2O. So it's a little confusing how the pH scale is derived, but a pH scale that is completely neutral is a pH of 7, and that has to do with the concentration of protons. We're not going to get into significant detail about how they come up with that number 7, but remember that 7 is neutral pH. Now, if you want to get a more basic solution, then it would be greater than 7, okay? And the higher you go, the more basic it becomes. A typical pH scale is between 0 and 14. Now, the lower you go from 7, the more acidic it becomes. So a pH of 0 is extremely acidic, much more acidic than a pH of 5 or 6, right? And so on. So in a way, 11 is more acidic than 12, but 11 and 12 are both considered a basic solution because they're both above neutral. What gets confusing about the pH scale is that, remember we said that pH is defined by the concentration of protons present, and so the lower the number, actually the more protons that are present. So you can think of acidic solutions having more protons than basic solutions. Basic solutions have increasingly lower amounts of protons available in solution. And why this matters is, again, because protons and acidic and basic solutions can change the sort of chemical reactions in the structure of chemicals in a solution. So pH affects the structure and function of all macromolecules. Extremely basic pH, which means a low amount of hydrogen, or protons there, or hydrogen cations, means that the hydrogen bonds that are already present in nucleic acids, they begin to separate at high pH. Lipids, which are, if you remember, important for our membranes and intracellular organelles and different things like that, they start to become hydrolyzed. So they start to break apart, so our membrane stability goes down. And then something called the proton gradient, which is used for cellular respiration. This proton gradient, there's a difference in the concentration of protons inside and outside of the inner membrane. This gets interrupted. This is known as a proton motive force, and the proton gradient is responsible for producing ATP, which is, of course, what we know as the energy currency of the cell. And so what happens at a high pH, where there's very low concentration of protons in a solution, if you put a cell in there, you can imagine that this would disrupt that proton gradient or cause it to collapse. And also under low or high pH, this can dramatically affect the folding, structure, activity, and charge of proteins throughout the cell. And proteins are very important tools for all sorts of different processes in the cell. Here's a graph that shows pH on the x-axis and growth rate on the y-axis. And like temperature, we can categorize microbes based upon what sort of ranges they can grow in. And so we have three main ranges to worry about with pH, or three main groups. And that is the acidophile. So the acidophiles, they can grow somewhere between 1 and 5.5 pH. We have the neutrophiles, which grow in a relatively neutral area, definitely solidly over 7, but about 5.5 to 8.5. And the alkalophile, or lover of basic pH, from 7.5 to 11.5. So most bacteria are neutrophiles. Most environmental conditions are neutrophiles on planet Earth. However, there are many valued microbes that are acidophiles. Within an acidic environment, it can prevent non-acidophiles from growing, and a lot of acidophilic microbes are actually human-friendly. So this becomes really important in food preservation properties, such as fermented foods that have a low pH, as well as certain parts of the human body like the vagina and stomach, which maintain a low pH, thereby regulating the type of microbes that can be present in those environments. So here's an example of that. This is beer pH over the fermentation time. So if you were to take wort, which is unfermented beer, basically the sugar juice from the barley that is produced, and you were to add yeast to that solution, or sometimes they even use different types of bacterium to supplement the yeast, you can see what occurs over time. So those yeast, if we look at pH here, the yeast will actually change the pH over time. So when they start consuming those sugars, they start producing acids, which lower the pH. So this pH gets lowered quite dramatically, very quickly, and then it prevents other sorts of pathogenic microbes from, a lot of different pathogenic and spoilage microbes from growing there. And we can also see, here's the Total acid production, so total acid production, of course, naturally goes up over time. And we see alcohol percentage goes up over time. And so, yeah, that's kind of how a lot of our fermentation processes work. Here is a list of foods and the common microbial acids that are produced within them. So we see for foods, we start with yogurt, and they have, you've probably heard of lactic acid, which is a byproduct of lactic acid bacteria. For vinegar, it's acetic acid. For kombucha, we can have lactic acid and acetic acid as the main constituents. In beer, you can have lactic acid and acetic acid as the main constituents. Now, let's go back and talk about osmotic pressure. As we discussed with osmotic pressure, it can affect the stability of the cell shape and the membranes. And of course, this is going to be very important for cellular growth. So if you can remember, osmosis is the movement of a solvent. In most cases, that's going to be water, to equalize the concentration across all areas. So this is a semi-permeable membrane. It's supposed to represent kind of that lipid bilayer. And so here are water molecules, H2O, and it's going to cross over until you get relatively equal amounts of solvent to solute, whatever particles are floating in there. And that's called an isotonic solution. So here's an example of osmotic pressure that can occur. So in cells in hypertonic environments, so these are environments where whatever the cells are in, it has a very high concentration of solutes, or those small little ions and particles in it on the outside, compared to within this cell, within the cell's cytoplasm. And what occurs is the water wants to escape and push out, and this creates some osmotic pressure on that membrane from the water. And so what can eventually happen is so much water can rush out that inner membrane, also known as the plasma membrane, can shrink and shrivel, and it can go through a process called plasmolysis, which is lysis or destruction of the plasma membrane. Of course, naturally, this can greatly affect environmental growth, or microbial growth. So most cells require an isotonic environment to survive, which is an equal concentration of solutes inside and out of the cell in order to achieve a proper osmotic pressure. Okay, so checkpoint 1. Why does adding large amounts of salt to meats preserve them and prevent microbial growth? Is it A, bacteria dislike the taste of salt? B, salt is a protective barrier to the product? C, excessive salt can create a harmful hypertonic solution? Or D, excess salt can create a harmful hypotonic solution? Okay, eventually we would get to this. Let's discuss oxygen requirements now. So we have a lot of different type of microbes based upon their oxygen requirements. Microaerophiles are organisms that require just a wee bit of oxygen in order to do some sort of cellular processes. Obligate aerobes, also known as strict aerobes, they're required to have oxygen present in order to survive. An example of an obligate aerobe are you and I, which demand atmospheric levels of oxygen in order to complete our cellular processes, such as aerobic respiration. There are the facultative aerobes. These are not strict aerobes. Facultative aerobes can use oxygen when present, and they prefer to use oxygen for doing metabolic processes because oxygen is really efficient. You can imagine, humans and multicellular life, things get really complex and are really hard to do. And so we need to do things really efficiently. So we use oxygen in order to survive. But so facultative aerobes might prefer the presence of oxygen, but they have ways to survive without oxygen. Okay, on the other side of the coin, we have obligate anaerobes. Obligate anaerobes are like obligate aerobes in that they're strict anaerobes. They must not have any oxygen present because oxygen is reactive and it can do chemical reactions not wanted by the cell, and it doesn't have ways to negate that like you and I do. And so it requires the absence of oxygen. And now we have aerotolerant anaerobes. It can be easy to confuse aerotolerance with microaerophiles, but the difference is that aerotolerant microbes or aerotolerant anaerobes, they cannot use oxygen, but they can tolerate it when it is present. So remember, microaerophiles require a small amount of oxygen. So we'll see this later on in how they grow in tubes. But first, let's discuss what's the issue with oxygen. So oxygen can have a variety of toxic forms. And the reason why they're toxic is because they're very unstable. And so when they... Sorry about that. So as I was saying, oxygen comes in many different toxic forms. They are toxic because they are highly unstable. So when they bump into biomolecules and macromolecules within or on the cell, they can react with them and turn them into things that are less functionable. The first example that I'm going to list here is called superoxide anion. So an ion is a molecule or an atom that has a charge. So this anion means that it has a negative charge, and meaning negative in this case. And so it has an O2 negative charge to it. And this is extremely unstable. So it can react with another O2 here and a hydrogen here and create a O2 that's not an anion and a hydrogen peroxide, or H2O2. This process is catalyzed or moved along by an enzyme called superoxide dismutase. So this enzyme drives this reaction forward. And this is great because H2O2 is a little bit more stable than superoxide anion. However, you've probably heard of H2O2 or hydrogen peroxide. It is a toxic compound. We use it for an antiseptic. So it can be used in order to try and kill cells. So it still is relatively unstable and harmful. So cells that have catalase can catalyze this reaction where they convert H2O2 into 2 water molecules and an oxygen molecule. And I apologize, back here, this was supposed to be one line. This was supposed to say 2 hydrogens. So this is a superoxide anion plus another superoxide anion plus 2 hydrogens makes O2, or this is just the type of atmospheric oxygen we have, plus a hydrogen peroxide. But anyway, when we get down at this very end here, water is a neutral substance, and so is atmospheric oxygen. Okay, so remember these obligate anaerobes that cannot survive in the presence of oxygen? They can't survive in the presence of oxygen because they do not make the enzyme superoxide dismutase, nor do they make catalase. So they cannot get rid of these reactive species. And these reactive species just naturally occur when you have oxygen built up in an environment. So They just naturally form through as byproducts of cellular reactions. I'm sure some of you, or all of you, have probably heard of antioxidants. Antioxidants are considered healthy because they are substances that prevent the toxic effects of oxygen. There's a lot going on in this table. This is a representation of the different groups of organisms based upon their oxygen tolerances. Again, we have the obligate aerobes, facultative anaerobes, obligate anaerobes, aerotolerant anaerobes, and microaerophiles. We'll start one by one and work our way down the line. So obligate aerobes, meaning oxygen, they require, or they're strict aerobes, they require oxygen. And if you grow them in a tube without disturbing the tube, the bacteria that do survive will be grown at the top. As bacteria die, they lose the ability to float, and you will see some settling here. But you would ignore those cells that settle at the bottom. As you can see with all of the other tubes, you're not going to have something that grows really well with a lot of oxygen and grows really well with zero oxygen, and there's nothing in the middle. So if you see a layer of bacterial cells at the top, even if you see some at the bottom but nothing in between, you're probably looking at obligate aerobes with some dead cells on the bottom. Okay, so the reason for that is where there are high concentrations of oxygen that diffuse through the atmosphere into the solution is where you're going to find those cells. And the reason why they do so well is they have those two enzymes we talked about, the superoxide dismutase, abbreviated as SOD, right, and catalase. Okay, so the next one are facultative anaerobes. So you'll find their growth both in aerobic and anaerobic environments, but remember I stated that oxygen metabolism is more efficient. So you see it's a little bit denser here at the top. You're going to find a little bit throughout the rest of the medium. So again, they grow best in areas that have the most oxygen present, but can grow throughout the tube. And they have the presence of catalase and superoxide dismutase, just like obligate aerobes, so they can tolerate that high oxygen. But they also have other metabolic processes that allow them to survive in these less efficient environments with less oxygen present. And the third group are the obligate anaerobes. And remember, they're the other side of the coin to obligate aerobes. Obligate anaerobes are strict anaerobes, meaning they cannot grow in the presence of oxygen. So you're not going to find some at the top and some at the bottom. You will just find them at the bottom where there's no oxygen or very little that diffuses and reaches the bottom here. So they're protected more from the atmospheric O2. Again, these little guys lack the enzymes superoxide dismutase and catalase in order to protect themselves from the harmful effects of oxygen anions or its reactive oxygen species. Fourth on the list are the aerotolerant anaerobes. And so these ones, they don't use oxygen, okay? They don't have the special metabolism that allows them to use oxygen as an efficient form of metabolism, like our obligate aerobes and our facultative anaerobes. So you don't see that special clumping of cells at the top. Instead, you see they kind of look like facultative anaerobes, but they have no bias for an oxygen-rich environment. So there you see them kind of evenly spread throughout the culture. So they do have the presence of one enzyme, that's the superoxide dismutase, so they can get rid of that horrible, if we go back, that horrible superoxide anion. But because they lack catalase, they can have a buildup of hydrogen peroxide. And finally, we have the microaerophiles, which are kind of a strange case. So these guys, they require just a small little bit of oxygen. So they need to be in this Goldilocks zone. Not too much oxygen and not too little. And that's because too much oxygen is just toxic to them. They can't be exposed to normal levels of oxygen. What enzymes they have is a little bit more complicated. It depends on species. So you don't have to worry about memorizing every different species that are microaerophiles and whether or not they have this enzyme or that enzyme. And so again, you'll see them growing in kind of the middle of the tube. So I recommend making yourself a little table and a note card, and remember what you need to keep track of here. We have 5 different groups. You need to know, do they require oxygen to grow? Can they grow in the presence of oxygen? Right? You need to know where they grow in a solid growth medium tube. And so we have the top, the bottom, evenly throughout plus a higher collection on the top, or right in the middle. You need to know why that is. So do they have catalase or superoxide dismutase? Do they just have one, like aerotolerant anaerobes? Do they have none, like obligate anaerobes?
Checkpoint 2. Based on the growth pattern, what are the oxygen requirements of the microbes in tubes A and B? And I want you to note here that in tube A, the living cells are in kind of the middle of the tube, not at the top, not at the bottom. What you see at the bottom there, ignore that. Those are just dead cells. And for tube B, the living cells are really in the lower part of the tube. They got a little bit mixed up, but just assume they're in the lower part of the tube here for this question.
All right, let's talk a little bit about nutritional requirements. There are some basic elements that all organisms need. First, there's carbon. Then there's carbon, which is involved in basically everything. It defines what an organic compound is having compounds that are rich in carbon. Next on the list is nitrogen. Nitrogen is essential for amino acids. The amine group is a nitrogen group on amino acids, so it's involved in all things protein. Sulfur is also a compound that's in some of the amino acids that we have in our bodies, and in bacteria, of course, in all life, and so therefore it's also required for proteins. Next is phosphorus. Phosphorus is a component of ATP, adenosine triphosphate, DNA, and RNA. So it's important for synthesis of all of those molecules. And then we have a bunch of these other trace elements, like iron, copper, zinc, etc. And they're used for enzymatic reactions. They help enzymatic reactions take place. So they're called enzyme cofactors. We use all of these ingredients in order to make a culture media. A medium is the nutrient material upon which microorganisms grow. This medium can occur in either liquid or solid form, but it must be initially sterile, which means that it contains no living microbes, so that you can inoculate it with your particular wee little beasties and have a tube that is only growing what you want it to grow. Let's first talk about solid media and agar. So all media, whether it's a liquid or a solid, contains the necessary nutrients for microbial growth. However, solid media contains agar. Agar is a complex carbohydrate. It comes from kelp or algae, and it is used as a thickening agent for solid media. Agar is essentially the jello of microbiology. It generally is not degraded by most bacteria and is relatively low in toxicity, so it's a great source for hardening different sorts of nutrient broths in order to make a solid structure. So, as we can see here, this person is pouring some agar into a dish, and this is called a petri dish, and it comes with a kind of a plate to pour into, or basin, and then this lid to protect it from the outside air. Agar, if any of you have ever used gelatin before, it acts and behaves quite similar to gelatin, although it is more resistant to being biodegraded. So here is a pile of agar right here. It comes in this dry powder form, and it's mixed with your nutrient broth, and then it's heated. And usually this is through a pressure cooker, and we'll talk about that later, but it's heated to above 100 degrees Celsius. And once it's heated to 100 degrees Celsius, then those little solid particles melt. And so it melts above 100 degrees Celsius. And scientists will cool this down to a temperature that's that's easier to handle, usually around 55 degrees Celsius. And it remains a liquid. And so this scientist is pouring a gel, and this agar in here is still liquid, so it's probably around 55 or so. But once it goes into this dish, it'll be allowed to cool down to around 38, 37 degrees Celsius, or the temperature of the human body. And once it drops below that, it will gel in temperature. The fascinating thing about agar is it doesn't turn into a liquid again until you heat it all the way back up to 100 degrees Celsius. So once it turns into a state, it has to travel quite a ways before it turns into the other state. So again, this is really stable unless for some reason you were to heat it up to sterilize it all over again. Another very useful quality or trait characteristic of agar. So that's one of the things the scientists like about agar, is that it has a large temperature range that it remains a solid. Now this is a great prelude into talking about sterilization techniques. In order to sterilize, scientists often use an autoclave, which is essentially a pressure cooker that kills everything. And in order to accomplish this, scientists need to heat the solution up to 121 degrees Celsius for 20 minutes. As you'll notice, 121 degrees Celsius is past the boiling point of water, which is 100 degrees Celsius. To accomplish this, though, if you apply a pressure to the vessel, so everything inside of here, of 15 psi, and you apply heat, everything can heat up to 121 degrees Celsius before boiling and evaporating. It is a timed process, though. If you were to do this, if you were to heat up your solution to 121 for 5 minutes, it wouldn't do the trick. We really need a full 20 minutes of sterilization. Dry heat is also effective. They recommend 160 to 170 degrees Celsius for 2 to 4 hours. So you don't really have to worry necessarily about the pressure, because you're not trying to keep things in a liquid form here. This stuff is just sort of a dry thing, so that you can bake glass, labware. This isn't going to be good for producing medium, so no liquids for this. And the other way that scientists do it is through filtration. So if you use a filter that has very small microscopic holes, the size of the holes do vary, but in general, if you're trying to filter out bacterium, for example, or you might not filter out viruses, but if you're trying to filter out bacterium, which are reach kind of a minimum size of about 1 micron or 0.75 microns, the typical filter is a 0.2 micron. This can be good for media that can't typically be heated. So there are some nutrients like glucose that if you heat them for too long, they begin to break down and form compounds with other nutrients in the media. So a glucose solution would be filter sterilized instead of heat sterilized. There are two types of media, which is determined by the type of nutrients that are used in them. One of them is a chemically defined media, and this uses a recipe with defined and exact quantities of each component. So in order to have very precise and exact quantities, this would lack any animal source unpurified ingredients. So instead of using some sort of protein extract from an animal, it would just be this amino acid and that amino acid and the third amino acid in this concentration for each one and individually. And it contains strictly purified compounds like glucose and table salt, NaCl, iron, etc. Next are complex mediums, and this is far and away the majority of mediums that people use. And it's made using a recipe that can vary from batch to batch, and that's because the sources of ingredients for these batches tend to also vary in their chemical makeup. So they can contain extracts from meats, yeasts, or plants that serve as sources of vitamins and nutrients. Now we can take a look at the methods for growing microbes and how they correlate to the type of media we have. So we can take a chemically defined media, or we can also take a complex media and create either a selective or differential media from it. So a selective medium is designed to selectively grow one type of bacteria and suppress other types of bacteria. Therefore, it's selecting just one group of organisms. Differential bacteria is not designed to selectively grow one or another. Rather, it's designed to have some sort of a distinguished phenotype between one organism and another. So you can tell by looking at the colonies that grow on it. You can look at the phenotype of those colonies and tell the difference of one microbe versus the other. So let's take a look at an example of each. Here we have an example of a selective media. So this selective media is called bismuth sulfite. Bismuth sulfite inhibits the growth of gram-positive bacteria. It only allows Salmonella typhi to grow in the presence of bismuth sulfite. So it's used to isolate S. typhi from fecal samples. You imagine fecal samples would have a whole slew of different microbes in them, but here we see in this image we're only getting the S. typhi to grow. We're selecting for S. typhi. This is an example of a differential media. This example is called blood agar. So this actually has blood made as one of the components to it, so you can guess that this is actually a complex media. And species that are able to destroy these red blood cells, they cause those red blood cells to lyse open, and that changes the color of those red blood cells. And so the color goes from this rich red color to kind of this clear, we call it a halo, where it's kind of like this light yellowish orange color. So an organism that's not able to do this process would be identified as not being able to do hemolysis or the degradation of the red blood cells. And so that's an example of a differential media. This is an example of a media that can do both. So it's multitasking here. This is a differential and selective media. This is mannitol salt agar, also known as MSA. So first, there's a high salt concentration which makes the plate selective. Only salt-tolerant genuses like Staphylococcus will be able to grow here. Staphylococcus is an organism that's present on your skin, and you imagine your skin has a lot of salt, so we can get Staphylococcus to grow on this plate. However, this plate also contains mannitol, which makes the plate differential. So mannitol is a type of sugar, and added to this plate is a compound called phenol red, which turns yellow when pH drops below 6.8. And if you remember in that image I showed you of the wort fermentation with the beer, as the sugars get consumed, acids can be produced by certain microbes. So if this mannitol is a sugar that the organism is able to degrade and make acids from, it will change that phenol red from a red color, you can see in this uninoculated example, into a yellow color. So it is this medium, we can see it's selective and that it only allows Staphylococcus to grow, and it is also differential because Staphylococcus aureus has the ability to ferment mannitol, creating a phenotype of a yellow color here, whereas Staphylococcus epidermis does not ferment the sugar mannitol, and so the medium remains red. And this lighter color right here is just the cells growing on the surface. It's not actually changing the color of the medium beneath it. Next up is McConkey agar, which is this really cool agar which is differential and selective. It contains crystal violet and bile salts, and these two compounds suppress the growth of gram-positive bacteria. So we can see that it's differential for the selection of gram-negatives, or if it's, we can say it another way, it's differential for suppression of gram-positive bacteria. It has a pH indicator. It's not phenol red in this example. It's actually called neutral red, and this neutral red turns a hot pink color at low pH. Low pH here. So as you can guess, we're probably going to use that same trick where we add a sugar that only certain microbes can ferment or break down and create acids from, and that sugar is lactose. Lactose is added to the medium, and bacteria that can ferment it will lower the pH of the sugar and make this hot pink color present. So we can see E. coli, if E. coli can grow on McConkey agar. So it's a gram-negative bacteria that is lactose positive, and then Salmonella is gram-negative and it's lactose negative, and so it would be no hot pink. That brings us to our next checkpoint. Checkpoint 3. EMB agar contains eosin and methylene blue. Eosin is toxic to gram-positive bacteria. Methylene blue turns black in the presence of bacteria that produce lactic acid. What kind of medium is EMB agar? Selective, differential, both, or neither? Okay, here are a couple plates. This is EMB with E. coli. It's usually reddish. And the second plate is MSA with Staphylococcus aureus growing on it. Let's talk about fastidious microbes. Some organisms are fastidious, meaning they are picky in their nutritional requirements. An example of this would be Neisseria gonorrhea, which requires blood or hemoglobin that has special amino acids and special vitamins in order to grow. In fact, most organisms are so fastidious they can't even be grown in the lab. If you remember back to, I believe it's our first lecture, we covered that over 99% of microbes are non-culturable. No conversation about microbial growth would be complete without talking about generation times and doubling times and the population density throughout a growth curve. So as discussed, bacteria go through binary fission, and the rate at which this happens is somewhere between 20 minutes to 24 hours for most bacteria. Generally speaking, pathogens tend to multiply quite rapidly on this scale, so closer to the 20-minute side, where some other microbes that exist out in nature might tend to grow a little bit slower. So the generation time is the time it takes for you to go from one cell to a pair of cells. So a synonymous term for this would be called the doubling time. So you've gone from 1 to 2, you've doubled the number of microbes. So if we look at this little graphical depiction of bacterial cells dividing, we can see that there is a pattern that emerges from this. From one generation, we go from 1 to 2 cells. And now we have 2 cells that can each go from 1 to 2 cells. So we go from 1 cell to 2 cells to 4 cells to 2, 4, 6, 8 cells. And from 8 cells, another doubling time would create, that's right, it would create 16 cells. So each one of these doublings is called a generation. So if we start with our founding population, we're looking at generation 0. The first generation, or the first doubling time, is our n equals 1. And here, because we started with 1 cell and we double, then we have 2 cells. Our second generation is when that first set of progeny multiply. And so they go from 2 cells to 4 cells, where cells is 4. Finally, in this example, we have our third generation, when we go from 4 cells to 8 cells, or another doubling. We can draw this pattern as a formula up above if we just start with our generation 0 and write down the number of cells, which is 1. Now we're going to experience a doubling of that, so we're going to multiply that by 2, or a second generation. And we'll have a third generation here, so we'll multiply it by 2 again. Just imagine we're just drawing what we're seeing in these lines here. Another doubling. And then a third generation here is this second multiplication, or this third multiplication, apologies, at the bottom here. So it's 1, and our pair of arrows for doubling is times 2, and then 4 arrows, so we're going to times it by 2 again. And then 2, 4, 6, 8 arrows, we're going to multiply it by 2 again. So in our answer, we had 8 cells after 3 generations. That's because we take our founding cell, which is 1, and we multiply it by the number of generations. So times 2, times 2, times 2, and our final number of cells is 8. And we can extend this out as long as we want to. And what I'm going to do is derive an equation from this. So 2 times 2 times 2 is the same as writing 2 to the third power. That's how many times you're going to multiply it, right? And so we're going to write an equation on this next slide to represent this. Remember, 1 was our starting number of cells, right? So let's go ahead and take a look at this next slide here. So this is how we calculate cell numbers. So our starting number of cells times 2, because remember each generation we're doubling, raised to the number of generations is going to equal our final number of cells. So in this shorthand for it, we have capital N, which is the number of cells at generation 0. Remember in that previous slide, generation 0, we just had one bacterial cell. We're going to multiply that by 2 raised to lowercase n, which represents the number of generations. In the previous slide, this would have been 1 times 2 to the third power. Back here. 1 times 2, there's 3 of them, 3 generations to the third power. See, 1 generation, 2 generation, 3 generations. You don't count the first one. There's no multiplying here yet. It's only this part that gets multiplied. So let's see how that plays out. Let's provide an example here. Here's the example. You have 2 E. coli cells that were transferred from the cook's hands into warm Thanksgiving gravy. If E. coli doubles every 20 minutes in the gravy, how many will there be after 6 hours when it is served? Okay, so we know a few things from this, right? We know the number of starting cells. That is how many? 2. It's 2 E. coli. We know that we're going to multiply that by 2 raised to the number of generations. Well, that's where it requires a little thinking. How many generations have we seen so far? Okay, so it gave us some clues. It tells us that a generation takes 20 minutes, right? And that there are 6 hours at which this goes on for. So every hour we get 3 generations because 20 goes into 60 minutes 3 times. So 3 times 6 should equal 8 generations. I'm going to draw this, or I have this written down here below. So 1 hour is equal to 60 minutes per hour, right? And then we have 1 generation per every 20 minutes. So that equals 3 generations per hour. And then if we have 6 hours, we multiply that by 3 generations per hour, we get 18 generations altogether. So we set up this problem by saying our starting number of cells is 2. We're going to multiply that by 2. And we're going to raise that to the number of generations, which is 18. So if you plug that into your calculator, 2 times 2 to the 18th power, you get 524,288 E. coli cells in the final solution, that final gravy. In scientific notation, this is written as 5.24 times 10 to the 5th. So in scientific notation, you try and have one number to the left of the decimal place and the other numbers to the right of the decimal place, and then you raise everything to whatever 10 to the whatever power. So we see if the decimal place is after the 8 here, we count back. We count 1, 2, 3, 4, 5. So we had to move the decimal place 5 times to get 5.24. So it's 5.24 times 10 to the 5th cells. As you can see, it only took 6 hours to get to 500,000 E. coli cells from only 2 founding cells. That's why it's so important to make sure that you're not leaving things out in a warm environment for a long period of time. However, there are some microbes that grow very, very slow. So this paper right here shows that there are long-lived bacteria that are reproducing only once every 10,000 years that live in rocks deep in the soil, about 1.5 miles below the ocean floor. And they can be as much as 100 million years old. So microbes have a diverse array of survival strategies. And so in certain environments where nutrients are not always the best or environmental conditions aren't always the best, they just grow slower rather than dying. So I think what you'll find is I'm going to post a practice slide with several practice math questions that you can work through that will kind of help you answer these checkpoint questions if you need help. The equation is not too bad. You have the final number of cells is going to be equal to your starting population size times 2 raised to the number of generations. Let's jump into checkpoint 4, our first major math question. If an infection starts with a single bacterium that undergoes binary fission every hour, how many bacteria will there be after 5 hours? So unlike E. coli, this one only divides every hour, and your starting population is 1. So after 5 hours, do you have A, 16 bacteria, B, 32, C, 11, or D, 10? So what we're trying to get at here is to show that bacterial growth is exponential. Here we can see that the number of cells and then the number of cells expressed as a power of 2. Okay, so this is a visual representation of the numbers. If we have 1 cell, then it's going to look like 2 to the 0 power. 2 cells is going to be 2 to the 1st power. 4 cells is 2 to the 2nd power. 8 cells is 2 to the 3rd power. And then 16 cells is 2 to the 4th power. So this would be if we had 1 cell starting out and we do 4 generations, we're going to have 16 cells. And if we have 1 cell starting out and we have 5 generations, then it's going to be 2 to the 5th, and we'll have 32 cells here. And if we put this on a graph, this just shows the insanity of what a exponential growth curve looks like. So here are the number of bacteria, and you can see just how quickly these numbers begin to rise. So a species that doubles every 20 minutes like E. coli can go from 1 to 4.7 times 10 to the 21st cells within a single day. So bacteria are commonly grown in batches. A batch is just a method of growing in which no additional nutrients or compounds are added to the growth medium, and no waste products produced by the bacterium are removed in the process. So it's kind of like a whole start to finish. No microbes in the beginning. You inoculate with your microbes. They grow up, and then eventually nutrients decrease and waste products build up, and the culture slows and then eventually dies. And one way that scientists have in order to measure this is called culture density. So culture density, this is important, is defined as the number of cells in a milliliter. So it's crucial that you understand that the volume is a milliliter. It's not a liter, it's not any other sort of measurement but a milliliter. So in this batch culture, there are distinct phases that occur throughout the growth of the culture. And let's take a look at those different phases of growth. So here we have 4 different phases of growth. So I want you to imagine We have a sterile tube. We've made fresh media in it. It has all of the nutrients required for maybe a culture of E. coli to grow. And the culture is initially inoculated with a small amount of E. coli. And this is the log of number of bacteria. So log is just a way that scientists often record the number of cells of bacteria because they get rather large eventually. So it's a way to kind of see the curve of the bacteria. And so initially, when you first add those E. coli, it's kind of like you just drop them on a deserted island. They have to first kind of sense what's going on around them. They have to start making cellular machinery and components in order to grow and things like that. So you don't have a lot of microbial growth or cellular division in this first phase. So this is called the lag phase. So it's in a phase of intense activity, preparing for population growth, but no increase in population. The lag phase enters into the log phase. So this is a logarithmic, or also called an exponential growth phase, and that's what we saw with those doubling times. So if the doubling time always remains the same at 20 minutes, for example, with E. coli, then you see this log phase right here where the population takes off in this linear growth on a log curve. This is called a semi-log curve because only the y-axis is in log form, whereas the time is not. So eventually the log phase leaves and the bacteria or the microbes enter a stationary phase, which is a period of equilibrium, so the number of microbial deaths balance the production of new cells. Eventually, though, we hit the fourth stage, the final stage, which is death phase, and this is where the population is decreasing at a logarithmic rate, so it drops off quickly as well. So the log phase essentially stops when the population approaches the limit of what the environment can support in terms of how much nutrition and fuel is available. There are many ways of measuring microbial growth, and we're going to focus on two main ways, which are plate counts that use serial dilutions, and then a direct microscopic count. The most common method for directly quantifying microbial growth is actually referred to as plate counts, where you actually count the number of what are called colony forming units on a plate. So each one of these colony forming units is not just one bacteria. Remember how big this plate was in relation to the person's hand when they were pouring that agar plate. What these represent are, on average, a single bacterium that lands on this spot on the plate and makes an entire colony out of it. So you're looking at thousands and thousands of bacterium in each one of these colonies. So that's why they're referred to as a colony forming unit. And so scientists would assume that there's roughly one bacterial cell per colony. So if you were to count the number of colonies on this plate, you would know that per volume that you poured onto this plate and spread around, there would be that many colonies. So if you counted 67 bacteria, there would be 67 bacteria per whatever unit of volume you poured onto the plate. There's a problem with this though. Their bacteria can be so numerous so quickly that it's very easy that if you were to pour something out of a broth tube and spread it around on a plate, that you would create what's called a lawn on the plate. So unlike that previous image where you could see those colony forming units, here those colony forming units are so close to each other that they don't form separate colonies, that they just form one giant mass on the plate called a lawn. So individual CFUs can't be distinguished and counts cannot be made. That's no big deal though, because scientists have a workaround for this. Um, Often in science, we have to dilute things several times before we can count how many there are. So in serial dilutions, we do just that. So remember that original broth tube that we poured onto this plate and created a lawn? Well, there is a method in which we could take that original broth tube called the original inoculum, and we can dilute it several times. So we could take 1 milliliter of that original inoculum and place it into a brand new tube of fresh medium, and then it becomes diluted. So if we were to put that fresh amount into 9 milliliters, then it would be 1 mil and a total of 10 milliliters of solution. And here we can see if we plated that out, you still cannot quite distinguish between different colonies. So let's say we try that again. Let's take this solution, we'll mix it up nice and even, and then we'll take another milliliter from that solution and stick it in another 9 milliliter broth tube. So 1 mil plus 9 mils means that it's now 1 in 10 mils, or it's been diluted so that there's only 10% of what used to be in this tube. And all right, so this looks a little bit better. We can start counting some of the individual colonies here, but still it's a little too bunched up. So let's do it again. We'll mix that tube up so it's nice and mixed, and we're going to take another mill and supply it into a 9 milliliter broth tube. And so that means now we have 9 milliliters of the broth tube. We added an extra mill, and that means we have 1 mil in 10 mils, or 1 to 10 dilution. And we can count these better, but there's still some clinging together, so we didn't dilute it quite enough. And so we'll repeat that process once again and again until we can find the right amount that they're easily countable and distinguishable from individual colony forming units. So we've diluted it down 1 time, 2, 3, 4, 5 times, where there's 1 out of 100,000th dilution, and we can finally count, oh, there are 3 colony forming units there. And don't be too stressed out about all the numbers here. We're going to go into this in greater detail in the future slides. As you can see beside each one of these dilutions, we have this ratio here. And this is the ratio of the amount of volume we added to the total volume of the new solution. Okay, so we added 1 milliliter to 9 milliliters, so the volume added was 1 milliliter and the total volume was 10 milliliters now. And from that we derive something called a dilution factor. So oftentimes in science, solutions need to be made. And these solutions can be made from a stronger solution in order to make a final concentration of the correct amount. Now when we have that final solution, sometimes we need to work back and figure out how much the original concentration was. Just as we saw in the previous slide, we would need to figure out what the concentration was in the original tube when we counted 3 CFUs on the plate. In order to do so, scientists use something called a dilution factor. A dilution factor is used to calculate the original concentration. The dilution factor is defined as the reciprocal of the dilution. So in this example here, we've made a very simple dilution of our espresso. We've taken 1 part espresso and added 2 parts steamed milk in order to make a latte. So let's think about this first intuitively, and then we'll understand how dilution factors work mathematically. So first of all, what is the percent concentration of espresso in the latte? You should have said 33%. There is 1 part espresso and there are 2 parts steamed milk. So it's 1 out of 3 is espresso, or 33%, 1/3. What fraction of the latte is the espresso? Well, I just gave you the answer. It's 1/3. Okay, so remember, the dilution factor is the reciprocal of the dilution. So what is the reciprocal fraction of 1/3? It's 3/1. All right, very good. So we can use this dilution factor to calculate the original concentration of the espresso. So what is the original concentration of espresso? So before it was made into a latte, what concentration was the espresso? Well, it was 100% espresso, right? And we poured it in that cup. We added 2 parts steamed milk. So the formula for calculating the original concentrations is the final concentration times the dilution factor. So the final concentration was 1/3 espresso, and we multiply it by the dilution factor, which is the reciprocal of the fraction, 3/1, and that's going to equal 3/3. 1/3 times 3 is 3/3, or 100% espresso. So that's a very simple example because the espresso didn't come from a diluted espresso already. It was already 100%. And we didn't dilute things by much, nor did we do serial dilutions. Let's take a look at another example. Okay, so in this example, we're going to look at how to calculate a new concentration. So we have 3 different flasks. We use flask A and flask C in order to make flask B. Flask A has 50% ethanol solution, right? So we're starting with a stock that's not 100%. And flask C is 100% pure water solution. So if we added 5 mils of flask A to flask B, and we added 5 mils of flask C to flask B, what would the new concentration be of ethanol? Well, if you think about it, you're kind of halving the concentration of ethanol, right? You're adding half of water and half of the ethanol to it. So it should be half of whatever this concentration is, intuitively. Let's see how it plays out when we actually ask the numbers here. So how much ethanol was added to flask B? Well, 5 mils. How much water was added to flask B? 5 mils. So intuitively, what do you think the percent concentration of ethanol is now in flask B? So we went from 50%, the concentration should be 25%. So what if we had to solve this problem in reverse? So now, let's say you know that you have a 25% solution of ethanol, and you know how much you added of each of these compounds. Can we calculate what the original percentage of ethanol is? Of course. Let's do this. So how much did we add of flask A? We added 5 mils. How much did we add of flask C? We added 5 mils. Therefore, what is the total volume of flask B? It's 10 milliliters. All right, so what dilution is this? Remember with the espresso, it was a 1:3 dilution, 1 part to 3 parts total. And remember in our dilution of our culture, it was 1:10, 1 part culture to 9 parts broth, which is a 1:10. So volume added over the total volume, which is 5 mils of our ethanol over 10 mils of the total volume, which is 5 mils of flask A and 5 mils of flask C. So 5 mils out of 10 mils, or we can simplify this and write 1 over 2. All right, so now let's create our dilution factor. So remember, our dilution factor is when we take the reciprocal of the dilution. So 1/2, if we take the reciprocal, we flip it upside down, and we get 2, or 2 over 1, same thing. Now we're set. So what was the original concentration in flask A? Remember, our formula is final concentration. In this example, it's 25% ethanol times our dilution factor equals the original concentration. So 25 times 2 equals 50%. So serial dilutions are a process of progressively watering down a bacterial culture to make it more dilute until you can isolate individual colony-forming units and then count those colony-forming units, and then use math in order to calculate the original inoculum. So let's rewind the clock. Now, say you take your original inoculum that the bacteria was grown in, and you want to figure out how many bacteria per milliliter are in that culture. So you know that you have way too many bacterium in this culture to count. So you decide to do a serial dilution. So you add 1 milliliter of this to 9 milliliters of a broth, and then you plate it. Okay, so your dilution fraction is 1 out of 10. 1 milliliter transferred out of 10 being the total. So the dilution factor is the reciprocal of that, which is 10. So if you were to be able to count all of the CFUs on this plate and say there was 100, to find the original concentration, you multiply it by your dilution factor, which is 10. However, you can see in this example that that's not the case. We cannot count all of the bacteria on this plate, all of the colony forming units. So this process is repeated again. So from this already diluted sample, you take 1 mil and you add it to another 9 mils and you make a new solution. Again, this is 1 mil added to a total of 10 milliliters because you have 9 mils there plus 1, so the total is 10. And so that again is another 1 over 10 dilution fraction. And so our dilution factor is again 10 here. So let's say you can count all these colony forming units, and there's 100 on this plate. So how do you find out how much was in this tube? You use your dilution factor. So you'd multiply it by 10, and that tells you that there's 100 times 10 or 1,000 in this one. And then you figure out how much there was in the original by timesing it by this dilution factor, which is another 10. So that's 1,000 times 10 is 10,000. So as you can see, we can go from a 1 to 10 dilution to a 1 to 100 dilution to a 1 to 1,000 dilution, a 1 to 10,000 dilution, and then our last one was a 1 to 100,000 dilution. So now let's reverse engineer how much bacteria we had in this original inoculum. On this last plate, we can finally count each individual colony forming unit. So we know that there were 3 bacteria per milliliter of this solution. But now we need to work our way back to the original solution. So if there were 3 bacterium per milliliter here, and we know that if we take 3 and we multiply it by our dilution factor, we can get the original concentration of this solution here. our dilution factor was 1 milliliter in 9 milliliters, or the fraction is 1 out of 10, so our dilution factor is 10. So 3 colonies per milliliter times 10 is 30. So we had 30 colonies per milliliter here. Now we want to calculate how much was in this one, and the dilution factor is the same. We diluted this culture 1 mil in a total of 10 mils. So 30 times 10 is 300. There's 300 colonies per milliliter in this tube. Times it by another 10, and we know there was 3,000 colonies per milliliter in this tube. Times it by another 10, and then we know that there were 30,000 colonies in this tube. And we know that the original inoculum was 300,000 colonies per milliliter. Now that's a lot of work. So we can see we had to times it by 10, 1, 2, 3, 4, 5 times, because we diluted it 1, 2, 3, 4, 5 times. Okay, that was pretty difficult. There's a way to simplify this, and that is just to multiply all of your dilutions. So we had a 1 out of 10 dilution, times our second one is 1 out of 100, times the third one is 1 out of 1,000, the fourth is 1 out of 10,000, and the fifth is 1 out of 100,000. So our dilution factor is 100,000, and if you were to take 3 and times it by 100,000, you get 300,000. Let's have a look at a word problem to kind of show how this plays out. So you grow a culture up, and from that culture you're going to do a couple serial dilutions in order to count the number of colony forming units you have. So from that original culture, you take 1 milliliter and you put it in a 9 milliliter broth tube. So what is the dilution factor with this first tube? You should have said it's a 10 dilution factor. And that's because there's 1 mil of your culture out of 10 mils total, and the reciprocal of that is 10. So then you take this tube and you take 1 milliliter and you put it into another 9 milliliter broth tube. So you've done another 1 in 10 dilution, or a dilution factor of 10. You plate 1 milliliter of this final tube and you grow it up and you count 25 colony forming units. So then the question is, what was the concentration in your original tube? So this is pretty easy. We're going to use our formula. We're going to take our final concentration of cells and we're going to multiply it by our dilution factors. We had 2 dilution factors here for 2 dilutions, and that will tell us our original concentration. So we had 25 CFUs, we times it by 10 for the first dilution factor, multiply it by 10 for the second, and we get 2,500 cells per mil, or 2.5 times 10 to the third cells per mil. Again, I will provide a little study guide for how to do these math problems. If you want to do them before answering these checkpoints, please feel free to do so. Okay, now here we have checkpoint 5. If the plate at the right represents a 1 to 10,000 dilution of the original culture, how many bacteria are estimated to be in each milliliter of original culture? So remember, you need to take your final concentration, multiply it by the dilution factor, and that will tell you the original culture concentration. And this answer will be in cells per mil. So we covered how to do plate counts. Plate counts essentially take a culture, you do serial dilutions, you plate those dilutions, and then you count the colony forming units. The colony forming units represent the number of live bacterium in that culture though. Not all the bacteria may be alive. And Also, it can be flawed because if two bacterium land in a very close proximity, or maybe they're stuck to each other and they land on that plate, they might grow as a single colony. So it doesn't perfectly represent the exact number of bacterial cells in that culture. If you want to count the exact number of cells, whether they're alive or dead or sticking together, you can use something called a hemocytometer. Hemocytometers were invented for counting red blood cells. In fact, heme for red, cyto for cell, and meter, so it measures the number of red blood cells. But microbiologists use these for measuring a variety of different types of cells. And what it allows you to do is count the number of cells under a microscope per a given unit of volume. So in this example here, we can see that there's the bottom slide piece, and there are these grooves here for aliquoting your bacterial suspension into, and then you have this cover slip, and this cover slip has a little bit of a gap between the bottom of it and where this grid is. So this grid has specific, very precise measurements of these etchings. So there's so much difference from this left line to this right line, so much difference from this bottom line to the top line of this square. So if you know all three dimensions, how wide these squares are, and the distance from the bottom slide to the cover slip, you know it has a known precise volume. So you can count the number of bacteria in any given number of cells, whatever is clever for counting purposes. You can take the number of cells and the volume that it makes up, and you can calculate, back engineer the number of cells per milliliter. So in this particular hemocytometer, you would take 0.01 milliliters of bacteria, and then you would spread that solution over the grid, and you would have this cover slip that goes on top of it. Okay, so there are also disadvantages for the direct microscopic count as well. One is if these organisms, microbes, are motile. Some bacteria, for example, are very motile. It makes it more difficult to be counted because they're moving around and you can't keep track of them very well. So they can move from one square to the next without you getting a good count on them. Sometimes you don't want to count the dead bacteria. So if you're doing direct microscopic counts, you're going to be counting the dead bacteria with the live ones. And it's going to be very vulnerable to sampling error. And that's because if you imagine with the plates, those colonies aren't going anywhere. They're just little cities that grow on that plate. And you can take your time and count them out. But in a liquid culture, it's really hard to count all of the cells because they're kind of floating around and moving around. And so from user to user, you might get different cell counts. And if the cell concentration is too high, you can again have issues of trying to count all of those individual bacteria. So even with this method, oftentimes you will have serial dilutions in order to dilute the culture volume into a countable number of cells under that grid.
Checkpoint 6. You view several 0.01 milliliter samples of bacteria using the direct microscopic count method and find an average of 126 bacteria per sample. How many bacteria are there per mil in the original sample?
Anyway, thank you so much for sticking through this one. I hope you all have good luck with working through those math problems. Go ahead and please review my math review document, and after you're done with those, please email me if you have any specific questions about the math. Thank you very much. I'll see you next time.